We're gonna get started. This morning we are super happy to have Mallar Chakravarty to tell us about structural magnetic resonance imaging. I guess some of you may already know Mallar. For those who don't, he's a prof at McGill University and working at the Douglas Institute. He's been doing structural image processing for all of his adult life, I guess.
Pretty much.
And it's in
particular. You can call me an adult now. Am I an adult already? Is it happening?
Slowly but surely.
Slowly but surely.
Yeah. Getting there.
I'm getting so big so fast.
But in particular, segmentation of region of interest in the brain through automatic alignment. So I'm going to let you take the stage. Thanks again.
Yeah, thank you. Thanks for inviting me. Can you guys hear me? Do I need a microphone? Is that fine?
Yeah,
can you hear me like that?
Yeah.
Okay. Oh no, but you're recording, right?
Yeah.
So you need a microphone.
Yeah.
Okay. So welcome and thanks for inviting me. So as Pierre said, I work over at the Douglas. I've been there for about 5 years now. And a lot of the work that we do, we do a lot of translational imaging work. I think for the purposes of this talk, I'm going to stick to what we do in the human. But actually, if you guys are interested in animal imaging, I'd be happy to talk about that too. More and more as our lab progresses, we're kind of a 50-50 animal imaging and human imaging lab. So what's kind of nice is a lot of the tools that I'm going to present today actually are very nicely translatable across species as well. There's a lot of work that we've done too. Again, I'm just going to stick to the human stuff though. So I'm going to talk about two pipelines that kind of have ended up being the workhorse in my lab. Almost every data set we run kind of goes through these pipelines. Again, there's not a lot of people here, so if you have questions, please feel free to stop me in the middle. The first is a very simple structural image pre-processing pipeline, yet another one which is probably just as good as a lot of the ones that are out there. It was created just to normalize operations in my lab, but what we found is that a lot of labs have also been using it across different, as a pre-processing stage across different image processing steps, and have found it to be very useful because it's quite flexible and it uses a lot of tools that you know well. And the other one is the maggot-brain pipeline, which I developed with Jason Lurch too long ago, and we use a lot now, and we're in the midst of turning that into kind of a deep learning based pipeline. But for now, I'll talk to you about the what exists and is available for others. So I don't want to convince people here why imaging is cool. Not just from the acquisition standpoint, we get better and better images every day. So on the left, if you can find a brain there, congratulations. That smudge was one of the first MRIs taken of the brain in Nottingham in 1978. And then if you fast forward 20 years, you have, or less than 20 years, you have these beautiful acquisitions, which are probably more, recognizable to you of gray and white matter structure in a simple T1 image. So this is the famous Colin 27 atlas where Colin Holmes went to the scanner at the MNI and the 1.5T scanner 27 times, and they just took a did simple linear registration and took an average of that brain to increase signal to noise. And then as you move on, I'm sure a lot of you guys are thinking about working in high field imaging with Human Connectome project data. And so you have these interesting problems where you have this exquisite data with incredible resolution, but now you have to figure out actually how to deal with it. So we almost sometimes, from an image analysis standpoint, image processing standpoint, almost lag behind our colleagues in the acquisition framework because they're kind of telling us, this is great stuff, you can do amazing things with it, and then you have to figure out how to then process it. So, well, the nice thing about structural imaging, I think, is that one, it's exceptionally reliable and reproducible, above and beyond most of the imaging modalities that we use. Not to say that you should use beautiful functional images or diffusion images, but more and more, I think we've realized we've learned a lot from structural imaging. The brain anatomy is far more dynamic than we ever thought it would be. So just if you think of development as its own trajectory, multiple imaging time points across a developmental trajectory from say age 5 to 25 can really tell you a lot about how humans turn into adults, like most of you. So the image processing pipelines can go from simple VBM pipelines to deformation-based morphometry, segmentation, cortical thickness pipelines, looking at the architecture of the folds of the brain by looking at the cerebral sulci. But all this requires some sort of common framework. Not all of it, which I'll go into today, but I'll talk about the stuff that comes out of my lab. So all the stuff we have is available online for free. You're more than welcome to go access it and download it. I can give you guys, they're either at the end here or I can give them to you later on. But they're freely available. I think what's also important to keep in mind, MagicBrain in particular is particularly computationally expensive, which I'll go into later on, and you'll see why in a moment. But what's nice about that is we've marshaled the compute calendar resources for these tools, for both these tools, and so what that means is that you can get a guest account on our kind of umbrella account, and we have a lot of wikis and stuff like that that'll guide you through how to run each of these pipelines. And then if you have questions, you just kind of ask the people in my lab about what's going on, and they can actually go in and help you directly in your compute calendar space. So I think that's a real advantage to using the tools that way. And you don't need a computer infrastructure, you just need your images, and that's about it. So it's all Linux based, they're all command line tools, there's no GUI or anything behind it, but you guys are smart and can figure that out. So the Bpipe implementation was originally used to work with Ming tools. I have to check in with Gabe Devenne in my lab, because I think he actually created an implementation that works completely in Nifty from end to end now as well, which is quite nice for everyone, because not everyone works in the Ming space. And then a lot of the other tools that we use are actually just based or ripped off of the Ants group, because we know and like those tools, and we've just kind of adapted them to do the work that we want to do. So there's a couple of things, and basically Bpipe is this kind of at the risk of being a big, self-deflating, it's a bunch of hacks that we realized work really well when you string them together and try to get your structural pipelines to work. So the first one is, when an image comes out of the scanner, it comes out in its own native coordinate space. And so, so that might be very far from something like MNI space, which most of us, or a lot of us rely upon in order to kind of align our data regardless of the analysis that we do, right? And so this could even be a pre-processing pipeline for your structural images, but in the ultimate goal of aligning your functional images, for example, or your diffusion images. And so it has this native coordinate space, and all we do is we force the center to be the middle of the image, and what you get is kind of some nice fake overlap between the original image and MNI space. And we can store that transformation as well, so that if you ever want to go back to the complete native space of your image, you can have that transform again. So we have a lot of options. Well, this is pretty easy. It's just kind of like a coordinate transformation of the image data, but it does actually help a lot of the downstream bias field correction, linear registration, nonlinear registration, and so on, because you have to do a lot less work to align your image to MNI space, or to align all your images together as well, if that's something that you're interested in doing. As you all know, bias field correction is kind of an important thing. This is an example from the Tutson paper where we use N4 correct instead of N3 in our case. N4 is kind of like a N3, which was developed at the MNI on steroids. It works really well. Gabe's created an implementation that does it kind of in a hierarchical scale space fashion. So he looks basically what we do is we marshal N4 and we process it a little more iteratively than they actually do. So for a lot of scans, especially at higher field or with interesting head coil configurations, you have the N3 or N4, they actually weren't created to actually find those bias fields that were there as a result of those kind of interesting hardware configurations. And so what we end up doing is we end up looking for low frequency bias field changes across the image, and then start kind of looking for more and more local changes over multiple iterations in the data set. And you can somehow control that, yeah?
Yeah, maybe some people have no experience in imaging.
Okay,
here.
sorry.
So maybe just because you can see.
Yeah, exactly.
Just explain really quickly what it does.
So effectively, when you want to drive your registration or you want to drive some sort of segmentation process, if you were to eyeball your image, you'd want the gray matter in image to look gray and you want the white matter to look white, right? And so what ends up happening is that because of multiple hardware considerations, so for example, if you look at that first one, the first column here, you see, well, this is a great, easy example. You have some bright spots and you have some dark spots, right? But ideally what you'd want is that for that to be a homogeneous image where things that are gray are grayer in the image and things that are white matter are whiter in the image, right? And what you can estimate is actually what's called that bias field, which is kind of that shady stuff in the middle. And that is the thing you want to remove from the image the bias that you want to remove from the image in order to create a more homogeneous gray matter representation or more homogeneous white matter representation. And when you do that, you get what's on the right-hand column. So actually, you have the N3 from the MNI, and you have N4 from the UPenn group. They're two bias field correction algorithms. Both show kind of the bias field that's taken away from the image, and then the corrected images in the second panel and the fourth panel, right? They're just two different ways of getting at it. We don't necessarily need to get bogged down in the technical details. N4 is basically a more current implementation of the N3 algorithm. And the idea is that, like as I mentioned before, it really starts looking for kind of overarching gross bias fields, and then starts trying to find more local inhomogeneities as it drills down, like you would kind of in a nonlinear registration algorithm, for example, right? Does that make sense? Any questions about that? This we found actually for a number of our applications, especially in cases where I'm sure a lot of you are thinking about doing this too, looking at public data. So for example, we have a recent paper using a big swatch of the Abide sample. And when we originally, and this implementation came out of that. And when we worked on that data set, we found we actually saved about 30 or 40% of our data just by implementing a nice bias field correction. So we then ran that data through Civit, which is a cortical thickness pipeline, which relies on gray-white matter contrast to really drive the cortical thickness estimation. We were also very strict with how we did things, but nonetheless, I would highly recommend this. Sorry, okay.
So I had a quick question. Is that sensitive to the brain mask? Because I know with N3 there's this sort of like chicken and egg problems that having a good brain mask helps correcting the non-uniformity within the brain.
So we can, yes and no. We do a few things now where we actually do a pre-classification, which is like quick and dirty, and we actually just constrain the N4 correction to the gray and white matter and try to, because you don't really care about the bias field in like, big ventricles, for example, right? You really want to kind of really concentrate your focus on there. And we found that really helps us a lot too. So we actually use, we don't use it for anything else. We just use it for the purposes of the correction, but we actually use the ANTS classification tool just to as a quick and dirty thing very, very early on in the pipeline. And then we focus our correction there. Does that answer your question?
Yeah. Thanks.
Yeah.
I was wondering, is that a kind of issue related to the strength of the field? Like in that with the 70 Tesla, maybe that will be less of an issue?
So these are actually, I think, 9.4 T images from this paper in particular. So the higher field strength you go, the more issues you have with inhomogeneity in general. Certainly 3 Tesla, when you start using faster switching gradients and more intricate head coil configurations, the bias field tends to be more focal in different regions depending on the transmit-receive characteristics of the head coil. So it's definitely, I think what's interesting is that N3 was created for like 1.5T data in the mid-90s, which had very kind of nice, smoothly varying inhomogeneity profiles, but we all want better resolution and better signal and better contrast, but that comes with other costs too, right? So that we have to account for.
But that's a common misconception, like higher Tesla is better. And for physicists, they love the higher Tesla, but from an image processing standpoint, a lot of things are worse at higher field, and all the distortions and non-uniformity are worse at higher field. So it's more challenging to process 70 Tesla images than it is to process 1.5 Tesla images. By kind of like a lot.
Surprising.
Well, it is and it isn't, right? Like, I think you have to, we went through the same thing. Like, I remember I was at the MNI when now I sound like an old fart, but the first time we got a 3T magnet at the MNI, I was there and I was a graduate student. And I remember that there was a significant amount of work put in to just adapting tools that we knew how to use in 1.5T and making sure that there was about a year or two of work that went into just making sure that we could either adapt or use these tools at 3T, and they would give us high fidelity across those two field strengths, right? It's not trivial. Progress happens, and sometimes people want to look at, say, local characteristics of the bold signal and using 7T, for example, right? And that might work really well for one part of the brain, but a great example is the HCP data, right? The Human Connectome Project data, some of the best 3T data ever required, and they've taken a lot of time to characterize this, but there's a lot of artifact in the medial temporal lobe area where a lot of people are interested. And I think, Pierre, you can correct me if I'm wrong, but a lot of the fMRI signal in there is not the greatest, right? Someone can correct me if I'm wrong.
What do you think?
For the ball signal?
Yeah.
I think it really pushed it too far.
That's extremely. Is this part being recorded still?
So
It's extremely diplomatic.
I had an interview with one of the HCP core members during HBM, and she was telling me about like how they push too fast. So I think this is not something that
It's not controversial.
It's not controversial. They try to accelerate the images as much as they could. They are the highest gradients in the world. They get excited, but maybe they went a bit too far.
But that's also part of science, right? If you don't try it, you don't. Until you collect 1,000 subjects, sometimes you don't know, right? Or you've already collected 200, and then, well, you got 800 left. So the twins are there. So yeah. So then we do, there's other steps related to affine registration. So what that does is that it just aligns each image, oops. Oh, it's supposed to be into the M&I space. It's supposed to be an animation here, which is not happening. But basically, it would align each image based on translation, 3 dimensions of rotation, overall scale, and the shear property into M&I space, right? So that's just it's called a linear transformation, which is fully invertible, and you can go back to your original image if you need to. Another thing that is so hacky and so trivial, and we thought, but we included it here, is that a lot of images, for some reason, are very generous with the neck and shoulder area. You know, I used to have a RA who used to say like, everything below there is just meat. I don't know if I agree with that, but for the purposes of image processing, you can treat it that way. And so we kind of treat it as table scraps, and we just basically do a nice cropping. We have a couple of ways to use our initial kind of registrations to identify where we should kind of, you know, safely cut off the neck of each individual to drive registration down the line, sorry. And this really helps our registration in M&I space, right? Does everyone follow me so far? Is anything here that it's a simple set of, you know, it's your most basic kind of image processing steps, but kind of strung along into one pipeline. We also have an implementation of Beast in there, which comes out of Louis Collins' lab, which is developed by Simon Eskildsen. It's a non-local means estimator to create a brain mask. We find it very robust. What's really nice about it is that it basically relies on a training data set. I think in the case of Beast, it used, I think, a lot of the ICBM cohort, which is young, healthy, normal individuals at 1.5 T. Others have added ADNI data. We've added some neurodevelopmental data, so it's quite easy to get it to grow pretty fast, and it's really adept at picking out kind of the templates in that library that are most like your image, based on field strength, based on morphological characteristics, based on intensity characteristics, and then kind of does this iterative estimation in order to create a brain mask. We find it a lot easier to use than at least the previous version of BET. I haven't played with the more recent ones. But because we can kind of tweak it to different populations, we find it quite helpful as well, and quite robust. We often use this as a pre-processing step also into, find it more robust at least with the constellation of tools I'm describing than some of the pre-processing tools that are available in Civit or FreeSurfer. And if we kind of give these pre-processed images in, like I mentioned before, we do save a lot of data, which is quite nice. Some optional stuff would be a non-local means noise correction. And so this is some really elegant work by Pierre Coupé when he was a postdoc with Louis. And basically what this work does, and you can basically see an original image that has very little noise in it with some noise added to it, some non-local means denoising, the absolute, you know, the image after the noise removal, and then kind of the noise characteristics that it's removed, right? We find this, now I'm gonna like wave a red flag here, we find this useful in some contexts and not others. In the case of cortical thickness, for example, it really shifts the boundary from before and after, so we don't use it in cortical thickness estimation at all. So I would, you know, a lot of our results that we have, you know, say something very robust, like you know, you take 200 people who are aged 5 to 85, you look at the cortical thickness as a function of age, this seems to make it disappear if we use this as a pre-processing step, right? For segmentations, however, we find it can be quite useful, and it can be quite useful, helpful. So it doesn't really shift boundaries that badly there. So just something to keep in mind. It's there if you want to use it. And the way it works is kind of similar to the previous BEAST estimation, where it kind of looks at a template library of images, and then it looks for kind of voxels that might be most like it and creates a weighted average and tries to find the noise characteristic across the voxels and remove them. So that's basically it for Bpipe. I think what's really nice about it in terms of flexibility is that depending on how you like to do things, a lot of these steps can be interchanged. You don't have to run them in any particular order, although we have a preferred order that we tend to run them in. You also find that because every data set has its own kind of unique personality, you might end up wanting to try different things with different data sets in order to get things to work. But that's kind of, it ends up, but I will say, like I said before, it does end up saving a whole lot of our data, and it's quite a simple thing to run at the outset. So any questions about that? And if not, I'll move on to Maggot Brain. Yeah.
Quick one, I'm sorry with that. I mean, thanks for the updates. You mentioned maybe a deep learning based version of that thing.
Not this yet, but we could do that. Although I mentioned a deep learning version of Maggot Brain, which is the next thing I'm going to present,
okay.
Which we don't have yet.
but you're not working on trying to adapt that.
We might. I mean, I think Louis is working on a lot of, some of those tools come from Louis' lab. But I think he's working on a lot of deep learning based updates for some of these. In my estimation, if we can. If we do like a segmentation pipeline where it's a deep learning base, you may not actually need any of these corrections anymore, right? Because if we train it with the original data sets, it should be able to figure that out on the fly if we're doing our job right. It might help out, but I mean, kind of like going from 1.5T to 3T to 7T, we don't know what, we still know what has to be tweaked when we go into kind of deep learning based segmentations and stuff like that. I don't know that anyone's really done that work yet. Do you have to do the pre-processing anymore? Like, do we care about registration for these kind of things? I'm not sure what the answer is yet. So I think we have, there's probably another 5 or it's not going to solve everything tomorrow, unlike the promise of many people, well, what many people suggest. I think, right? I don't know. Does that answer your question?
Yeah. Maybe for the audience, like some of the steps you've seen here, there's now some deep learning algorithms that do them well, like brain registration, for example, has state-of-the-art performance on some data sets. And it's also extremely fast. So it's really exciting. Like you can do a brain segmentation under a second.
On your laptop.
On your laptop, which means you can do it 100 times with a little bit of noise to see how robust it is, which is something we cannot afford with current tools because they're so slow. So I'm personally very excited for the next generation of image processing, which would run in a few minutes instead of a few hours.
Yeah. I'm completely with you on that. I'm sure my lab is as well, but I mean, maybe I'm being too pragmatic about it, but I'm usually like to kind of test these things out before we make the claims. I know there are some nice tools out there. Bpipe, for example, has some nice tools that are quite fast, run in seconds. And our goal is to kind of just have these deep learning based tools ready to go. If we can get our data sets to process in under a second or two compared to the multiple hours it takes now, that would be great. Yeah, so it's coming. I'm also very excited for it. I just want to make sure that we're putting out what we say we put out, right? Yeah. Any other questions? No. Okay. So next thing I'm going to talk about is our subcortical segmentation pipeline, and this is the one that I'm probably more excited to have as a deep learning application because you'll see why it's very expensive to run. It has a lot of redundancy built into it, which is why it works well, but also why it takes a whole lot of time and needs a computing cluster to run it. So Maggot Brain stands for multiple automatically generated templates. It's intended for hard to define atlases and hard to define segmentations, and I'll explain what that is in a little bit. And the outputs include volumetric segmentations and surface-based shape measures. So one of the reasons we came up with this algorithm is because I was working with this atlas in my PhD, which is derived from serial histological sections, which are here on the left, and they were reconstructed to form a volume, which we then warped to fit an MRI template, which we can then use. So once it's on an MRI, you can use it as an input to any segmentation algorithm. So this is actually like chapter 1 of my PhD thesis, and I've been carrying it around with me ever since. But then as I moved on in my career, we moved on to other interesting anatomies like the hippocampal subfields and the cerebellar lobules. And so the outputs of this include segmentation. So we have 3 sets of segmentations that you can get outputs for. One is the subcortical structures, like I showed before, the subfields, or the whole hippocampus, and then the cerebellum. And you can kind of mix and match these segmentations however you'd like. We validated them across different field strengths and different resolutions. So if you're interested in some of that, I'd be happy to talk about it too. And the other thing would be that we provide as output are these surface-based measures, which kind of give you more localized measures of the morphometry. So, one way to think about it is there's 2. One that's just kind of based on the unit normal here, where you're just basically seeing how far relative to the group average, how far does one individual travel at each point in space along that new unit normal outside or internally, right? So you have a little bulge there, or a small indentation there, right? And we've actually been working on the neurodevelopmental basis of these shape characteristics and the heritability of these shape characteristics, and it's actually quite interesting. They actually are quite heritable, but regionally, there's regional specificity to all this, which if you're interested, we can go over later. The other one we look at, which is actually probably more of a nice developmental phenotype, is the area expansion and contraction. So looking at the surface area expansion and contraction across each point in space as well. And so that's another nice feature. So you kind of run the pipeline once, but you get not just volume, but other kind of more salient measures that you can use. And I'll show you kind of some of the applications of those at the end of this presentation. So I guess Pierre mentioned that some of you may not be completely aware of what segmentation is, but basically if you have an atlas where we've defined the original anatomy and you have a subject that you're interested in segmenting, the very classical segmentation is to estimate a nonlinear transformation that matches the original atlas where the anatomy was defined to the new image, and then you warp the label set onto the new atlas, right? So for the purposes of the rest of this talk, we're going to turn this into like an engineering cartoon, and a label or a piece of anatomy with that little flag in the middle of the box. A brain will be a square box. The squiggly line will be your image registration algorithm, the nonlinear registration, and then on the right you have the image that you've segmented. So this has worked well. This worked well for a number of years, but you can imagine that just by using a single atlas, you have atlas segmentation errors, registration errors, or sampling errors that you just can't really overcome with a single atlas, right? And so what we've done is we've just basically used what's typically defined as a multi-atlas segmentation framework, where sometimes you use anywhere between 30 to 80 or several hundred atlases, kind of in an atlas library, do multiple nonlinear registrations to your subject, and then find some sort of rule for fusing the resultant labels to minimize the amount of error that you have at the final segmentation, right? So here's an example of that classical image segmentation algorithm where you have 30 or 80 of these atlases, and then basically in this particular implementation, the algorithm picks, I think, the 20 hippocampi that are closest to the hippocampus that you're interested in. It performs nonlinear registration of those specific subjects, after which you get a segmentation after the label fusion. So this is nice if you have 30 or 80 nice manually segmented atlases. We don't, because we have one set of serial histology and we don't have a lot of volunteers to give us more. And so we wanted to find a way to deal with this kind of unique property. So what I'm showing you here works well in human data, in rodent data as well. It really does improve the quality of our segmentations as we move through. So basically what we do is we just create a lot of pseudo-atlases from the subject that we're trying to segment. So here we start with 1 to 5 atlases. Now I think all implementations start with 5 atlases or 10 atlases. We've kind of reviewed, updated a lot of the atlases that we've been working with from the anatomy standpoint. We label some of the subjects as templates, and I'll talk about what that is, and we use these templates to guide the segmentation. So in our cartoon version again, what we do is we have the atlases here on the left. We take out a subset of the subjects and call them templates. And then we warp each of these atlases to each of the templates. So we artificially grow the number of candidate labels you have, and and the number of definitions you have for each individual subject. Then each of these templates then gets registered to a single subject. And so again, you artificially grow the number of candidate segmentations you have using the data that you have on hand. It's almost like you're training your data, your algorithm through the data that you're working with on the fly, and then do a label fusion after that to get the final segmentation. So in the case of subcortical structures, we've used this algorithm in a number of contexts. I'll give you a couple of examples. This is some work we did with Armin Rasnahem at the NIMH a number of years ago, but just looking at subcortical development as a function of developmental age. So in blue here you have boys, in red here you have girls. Each of the small points is an individual connected in time by the lines between them. And then the thick lines are kind of the group trajectory as a function of age, and the big dots are the peak volume detained, which is kind of a neurodevelopmental milestone attributed to in a lot of developmental work. So a couple of things that we noticed here that were quite interesting is that if peak volume detained is kind of like this developmental milestone, if you compare it to, say, cortical maturation, although those curves may be a bit controversial now given what we know about development, it might be straight lines kind of just going downwards, but nonetheless they're pretty close. What we do see is that the peak volume is attained, especially in the striatum and the thalamus, quite a bit later than what you would imagine in the cortex, right? So there's kind of this protracted development in these structures that maybe goes on for a little while longer. But if we're being, diplomatic about it, there's probably not a lot going on until after the age of 10. There's not a big change in the volumetric trajectory until after the age of 10. So one of the things we were able to do is we were able to define these shape characteristics based basically on surface representations of structures that we're interested in, and following the nonlinear transformation back to kind of an average representation of the group. And so what you do is you can get a surface representation like this, and you can, in this case, we're looking at the areal expansion, so the surface area expansion of each of these structures. Red is an increase in surface area, and blue is a decrease in surface area. In each of these cases, this holds if we normalize by total surface area or total structure volume as well. And so these are the unthresholded maps here on the left. In the middle are significant expansions as a function of age and significant contractions, right? So if you were just to look at the volumetric measure, you would be led to believe that there's nothing going on morphometrically in these structures throughout the developmental lifespan, right? Or the developmental part of the lifespan. But there is some sort of rearranging which is very specific to different parts of the thalamus. So if you kind of map this out into one of my favorite movies, you get something that looks like this. So here, red are regions that are, again, growing on the surface. And this is nice. We've looked at how well this correlates with local volume measures, and it's quite nice, actually. And then blue are the regions that kind of contract as a function of age. What's also nice about this, and we've kind of gone on to prove it in other data sets since then, is the regions in blue are kind of nicely preferentially connected to one another in the subcortex, but also nicely connected to like DLPFC regions in the frontal lobe, right? So just there is some sort of developmental kind of nuance to this. We've also applied this to other data sets, kind of that are more neuropsych that represent neuropsychiatric disorders. So this is in collaboration with Drew Rappaport and Jay Geed, and then in Gogte at the NIMH. And so they have this beautiful data set looking at what's called childhood onset schizophrenia. And this is a type of schizophrenia that has a very, very early age of onset. These kids have, really severe schizophrenia-like symptoms, by the age of 13. They're very hard to treat. They're usually pharmacologically resistant to most treatments, so they kind of go through this course of polypharmacy throughout their whole life. But it is very severe, and it really does kind of mirror what's seen in the pharmacologically resistant adult onset schizophrenia as well. And so these kids, if you kind of focus on the bottom line here, if you look at their cortical thickness, which is just the thickness of the gray matter ribbon at the top of the brain, throughout for the ages of 12 to 24, you see, changes in loss of gray matter, but it's not exactly a static fixed difference. It kind of changes and has preference throughout the frontal cortex throughout the developmental lifespan. And now schizophrenia is interesting because we know, like a lot of neuropsychiatric disorders, it's about anywhere between 40 to 70% heritable, depending on which study you choose to look at. And so an elegant thing that they managed to do through this data collection is they collected data from unaffected siblings, also longitudinally, to look at do these unaffected siblings who are otherwise psychiatrically healthy have some version of this brain phenotype that their sicker siblings do. And it turns out that they do, but that it's far less severe, obviously, and that signature goes away later on in life. So what we wanted to do is use the subcortical mapping as a function to look at this cohort as well. And so these are just the parameters. We had about 600 scans across about 300 individuals, all collected on the same scanner. And in this case, instead of using that aerial expansion, I mentioned this kind of surface-based morphometry metric where we look at how much people bulge or indent at the surface of the brain, and so that's the metric that we used here. And so what we found was that you see these kind of surface bulges. So if you look at it volumetrically, all you see are that the siblings and the normal controls have no difference, and that the kids with childhood onset schizophrenia have this monotonic increase in volume. I should include that plot here. But that increase in volume isn't terribly surprising. It's probably related to administration of antipsychotic medications. We know that the striatum swells quite a bit, especially with accumulating dose, right? So you can't really tell much from just looking at a volume measure here. If we normalize for volume in every single individual, their own volume or their surface area or any number of metrics, you get this similar pattern that emerges where you have this kind of bulging that happens at the back end of the striatum. This is kind of a weird view of the striatum. It's as if you're lying down and looking at it from like behind. In red, you have the childhood onset schizophrenia kids, their siblings in green, and the normal controls. And what you see very nicely is that in this shape measure is that the siblings sit right between the childhood onset schizophrenia group and the normal controls. And this is true if you look at the other way too, where you see this kind of indentation. On the bottom is the siblings, and on the top is the relative to the controls, and on the top is the childhood onset schizophrenia group relative to the normal controls. And you get the same kind of pattern that emerges here. And what's also interesting with this phenotype, like the cortical thickness phenotype, is that it kind of disappears in the siblings as they get older as well, right? So it's this very dynamic phenotype that changes as a function of age. And I think I'm going to stop there. So just some conclusions. Image preprocessing is great. You should do it. It'll help your downstream outputs. And then maggot brain can really help you with your volumetric and morphological outputs for non-cortical structures. A lot of this stuff can be found here. And I'll give the slides to Pierre so you can go and look at us. But if you look up Cobra Lab, a lot of this stuff is linked up off our lab website. And with that, I'll be happy to answer any of your questions. Thanks a lot. Yeah.
Very interested in this maggot brain software that you developed.
Yeah.
So my lab runs out of the MNI, sorry.
Yeah.
And we use Civet, and I was wondering how would maggot brain work? Would we run things through Civet and then maggot brain?
Yeah. So what's nice about it is the surface-based stats are basically the metrics on the surface are all output in the same format that Civet outputs its stuff in. So it's object files and then little text files that map on to the object file. It's pretty simple. And so you can use whatever you do to do your Civet stats, like SurfStat or RMINC or whatever it is that you use. You can use those to run your maggot brain stats too. So whatever you've learned to Civet, we made it kind of compatible in like the MINC world to do the maggot brain stats as well.
All right, so if there's no other pressing question, we're maybe gonna stop there. So just a few things. Mallar is kind enough to stay for about an hour or two.
Yeah.
So if you want to discuss with him, please try him.
Yeah.
And I know that if you want to use maggot brain or you have questions on structural imaging, I think in the coming weeks you can contact Mallar.
Yeah, for sure.
There's Gabriel, who is very, very helpful.
Yes.
So that's kind of our structural imaging branch. In an ideal world, you would have gone through all of that in week 1, but you know, week 1 has only so many hours.
I also wasn't here, so you can blame me.
It just didn't fit the schedule. You know, we had too many things scheduled. You'll hear about quantitative MRI next week, but I think at least during the duration of the BrainHack School, you're going to have a nice overview of all the main modalities and approaches. Another small announcement is that tomorrow we're starting the project presentations. And so for the past 2 days, it was quite laissez-faire. You were like exploring, thinking, it was great. But now it's time to start delivering something. And none of that is binding, you know. You still have 2 weeks to iterate and refine, but it's time to at least make some plans. So you need to write a markdown description of your project, create a repository, start asking for feedback, and try to formalize it through the GitHub issue system. All of those are really kind of a game for you to get familiar with those mechanisms. You can obviously go just chat with folks, it works too, but it doesn't scale. Outside of the BrainHack School, you won't have all those folks just sitting around you all day. So it just try to play the game and do those little things. It also helps for me grading, because then I just have look at the repo and be able to say, oh, they've done that, so I can give the percent grade. So I'm going to come nagging you throughout the day to make sure you're on track. And also, if you feel stuck and things are not clear, you really please reach out to me, Basile, Alexa, Mallar when he's here, Claire, or anybody to try to get going and get unstuck. So if we're all good for the maggot brain presentation, thank you again.
Thanks.